So our today's speaker is Dr. Lauren Webb, who is an associate professor of chemistry at the University of Texas at Austin. She completed her PhD in chemistry at the California Institute of Technology in 2005, working with Dr. Nathan Lewis, studying chemical and electronic properties of silicon surfaces. Then she moved to a postdoc laboratory of Dr. Stephen Boxer in the Department of Chemistry at Stanford University, where she was focusing on quantifying electrostatic fields in proteins using vibrational Stark, should I say in German, effect spectroscopy. Then Lauren Webb moved to University of Texas at Austin in 2008, where she, since the time she's developed a program on which is focusing on both surface and biological chemistry, which is her expertise. So let me start. Let's give the floor to Lauren so she can start, and I will start sharing the screen in a moment.
Well, while you're pulling that up, just want to make sure you can still hear me.
Yes, it's perfect. I can.
Great. And I just want to say thank you for the introduction, and thank you for the invitation. I think that we have a lot of research interests in common, and I'm really quite happy to be talking here and getting to know some of you a bit better in talking about our work. You know, I gave a very vague title. You know, a lot of our work right now on trying to understand the behavior of proteins, and particularly protein-protein interactions, is really starting to delve into the environment that we find the protein in, right? Whether it's a very crowded cell, in the case of what I'm going to be talking about today, the lipid bilayer membrane. These environments set up electrostatic and electrodynamic fields that then govern the behavior of the biomolecules. And so I'm going to be spending a lot of time talking about that today, particularly in the context of lipid bilayer membranes. And the proteins that I'm going to be talking about are pretty small. One's even just a single amino acid, but you know, we're working our way upward. Yeah, thanks. I think this will work out fine. Before I start talking, I just want to take the time to thank the people who've done the work. This is a relatively recent photograph of my group. Happy that we're out of coronavirus separations. The work that I'm going to be talking about today is really quite an ongoing story that started several years ago now by some former graduate students, Rebecca Shrestha and Carrie Anderson. And then carried on by Molly Kogan, who graduated much more recently and is now being done by Chad Drexler, postdoc, that guy with the beard in the bottom of the picture. And the work that I'm going to be telling you about has been done in collaboration, a very fruitful, really quite stimulating collaboration with my colleague Ron Elber, who's a computational chemist here at UT, and his postdocs Alfredo Cardenas and Armand Fatazeda. And I'll be talking about their work in quite a lot of detail later. And I just want to thank the various funding agencies who supported us over the years. So you can keep going. So by way of a very broad introduction to my lab in general, we're interested in what I would call the physical chemistry of biological interfaces. We're interested in understanding at a molecular level how large, complex molecules can interact with one another in very specific ways for conducting function, for building structures, etc. And doing this in complex environments, as I said, like the interior of a living cell, which is quite crowded and quite dynamic. And we study these problems from a variety of perspectives. We're very interested in understanding protein-protein interactions and how electric fields that are generated at protein surfaces can lead to very specific interactions that cause specific biomolecules to dock in a particularly effective configuration for carrying out some function, perhaps chemical catalysis, and yet avoid all of the other non-productive interactions that the protein is no doubt encountering in its life cycle in the living cell. We've become interested in recent years, more interested in trying to understand how electrostatic fields generated within a lipid bilayer membrane govern proteins that are associated with that membrane in some way. And I'll be talking about that quite a bit today, so I'll just leave that there. We're very interested in taking what's sort of on the left side of this slide, learning from nature, and applying it to over on the right side of the slide, new environments, integrating biological function with synthetic or inorganic or polymer materials to generate materials that can co-opt the function of an interesting biological molecule, but in the context of an environment for being used as, say, a sensor or catalysis for a practical application. And so if you go on, so because I'm going to be talking about lipid bilayer membranes today, I just want to briefly introduce why this is such an interesting environment for us to study. And this is a picture I like to show at the beginning of my talks. This is just a beautiful artistic rendering of a cell that you would find in a biology or biochemistry textbook. And I really like this picture for a couple reasons. First of all, it reminds us that when we talk about lipid bilayer membranes, we have to remember that there are many different membranes within a cell. There's, of course, the plasma membrane, and then organelles within that cell then have their own membranes. And the field of lipidomics, largely led by the mass spec community, has in the last decade or so cataloged chemical differences in these membranes in really quite a lot of detail. And we now understand that they're chemically quite different in ways that can be used to understand function, and then even further for example, targeted drug delivery to one specific organelle in one specific cell. The other thing that I really like about this picture, if you go back, is that the plasma membrane in particular is kind of transparent in this artistic rendering. It's the thing you look through. And I like to remember that because I certainly was taught, as an undergraduate, that the membrane's just kind of there to hold the whole thing together. And the membrane itself is really acting as sort of a barrier or a physical platform, but it itself isn't very interesting. And of course, that couldn't be further from the truth. And that's what I'm going to be talking about today. So if we go to the next slide, this is another way of thinking about the lipid bilayer membrane, where we're starting now to remember that it's chemically quite complex. So there are thousands of lipids in mammalian cells. And like I said, different membranes within a cell have different lipid compositions. There are, of course, many small molecules associated with a membrane. I'm going to be talking about cholesterol, but it's one of just many, many biologically relevant examples. And then, of course, there are proteins that are associated with in the membrane in some way. So they're either intercalated into the membrane, they're tethered to it, perhaps they're anchored to it. And these chemical components aren't just homogeneously distributed throughout this two-dimensional structure. There's now quite good evidence that they heterogeneously associate into domains, sort of at the bottom part of this figure, which are enriched in some molecules and exclude other molecules. And these domains appear to give cells function that, or the membrane function, excuse me, that, like I said, is far more important than just being a barrier. So if we go on, the other thing I want to keep in mind as we start talking about lipid bilayer membranes right at the beginning is that in sort of the cartoons that I've showed you up till now, it's a very traditional view of a membrane where it's really just a slab of a hydrophilic, hydrophobic, hydrophilic sandwich type material. But we have to remember that structurally it's far more complex than that. First of all, there's water. And so this is a snapshot of a molecular dynamics simulation done by my collaborator, Ron Elber. And I'll get back to this specific picture later. But here I'm just throwing it up to remind us that there's a heck of a lot of water, and it's not completely excluded from the membrane. It's, of course, in the head group region where there are charges But water can actually intercalate quite deep into the interior of the membrane. And sometimes when it does so, it can enable a mechanism for getting something into a membrane which wouldn't want to go in otherwise. And I'll show you a specific example of that. So this is chemically a very complex environment, and we want to understand what happens when we put another complex object, a biomolecule, a protein, or a peptide, as I've shown you here, into this milieu. So if you go on, so the question that we first started asking ourselves several years now, we had a longstanding interest in electric fields in biological molecules like protein, really going all the way back to my postdoc work. And so if you just draw out a simplistic picture of a lipid bilayer membrane, like I'm showing you here, where really we are just thinking of it as a sandwich with sort of a charged or hydrophilic head group, and then an uncharged low dielectric constant fatty acid interior, you can just pencil and paper estimate that this structure will generate some very interesting electric fields. Okay, and so we normally think about 3 electric fields associated with the membrane. And the first, and by far the best well-studied, is the transmembrane field. And this is the electric field that's generated when the compartment on either side of the cell has a different pH or ionic strength or something like that, and generates an electrostatic field just through that gradient. And of course, the neuroscience community has studied this in quite a lot of detail because it's very important for, for example, signal translation by moving ions across the membrane. Okay, so the general magnitude of this in human or biologically relevant cells is well known. If we go on, there's a second membrane that I'm going to call a second field, excuse me, I'm going to call the surface field, so F sub S, that's completely associated with just the head group region of the lipids. And you can think of this very much as you would think of the gooey Chapman layer of ions that have lined up on the surface of an electrode. These are ions that are lining up near this charged or polar head group in an organized fashion and generate quite a large electric field, but one that drops over a very, very short distance. And again, this has been well studied on, say, supported lipid bilayers, using very much the techniques from electrochemistry to study the gooey Chapman layer. But there's a third electric field that, again, you can just write out in pencil and paper that I've shown here. And I'm going to call this the dipole field. So I'm going to give it the symbol F sub D. And over here on the left, I have a DOPC lipid. So this is a phosphocholine head group, Zwitter ionic, and then the polar glycerol linkage eventually leading to the hydrophobic fatty acid. And what's important to remember, of course, is that there's all this water around the head group region. In fact, if you do just very simple molecular modeling, you can fit somewhere in the vicinity of about 10 to 12 water molecules around each individual lipid head group in a fully formed supported lipid bilayer. So there's a ton of water that's associated with the top part of this lipid structure. And it's not water that's randomly oriented. It's there because it's hydrogen bound to the Zwitter ion and to the glycerol. And so what happens is water itself, of course, has a fairly large molecular dipole moment, but now you have this collective dipole moment from all of these ordered waters pointing roughly in the same direction into the low dielectric constant interior of the membrane. And this generates a field. And because it's thought to originate from this organized dipole moment effect, it's called the dipole field. So that's why I'm going to give it the symbol F sub D. And unlike the other 2 fields, when we first got started several years ago, there'd been a lot of interest in this, but very little, essentially no direct measurement of the magnitude of this field. People had done a lot of indirect measurement, studying, for example, the rate at which ions are transported across the membrane, doing scanning probe microscopies, that kind of thing. And of course, it can be estimated from molecular modeling. And through that, there was kind of this agreement that this field was probably on the order of about 1 to 10 megavolts per centimeter. Okay, so that's an interesting range for a couple of reasons. One of which is it's huge, particularly compared to the two fields that we know a lot about. It encompasses much of the interior of the membrane. And by that time, by the time we started this work, I'd say about 2015 or so, there'd been really good measurements from my group, from my postdoc advisors group, Steve Boxer, from others, of course, that were more or less settling on this range, about say 5 to 10 megavolts per centimeter, as being electric fields that are generated by the structure of proteins themselves. And so if you're looking for a mechanism that is going to allow a protein integrated in some way into a membrane to move in a directional manner, to, for example, associate with some molecules and not others, it seems like this field would provide a driving force that's exactly the right energy that we would need. Okay, and so we were very interested in studying this dipole field directly. So this next slide is just the integration of all three fields here. And I just like to point out that even though I'm really going to focus in this talk on the dipole field, we have to remember, of course, that for a molecule that's interacting with the system in some way, it's not like it thinks to itself, you know, okay, now I'm in the transmembrane field, now I'm in the surface field, now I'm in the dipole field. Like, this is a complex energy landscape that changes quite dramatically as a molecule spans this structure. And so really what we need to do by focusing on just one specific aspect of this, the dipole field, is of course to learn how to integrate it with everything else that we know about electronic arrangement and structure of the system. Okay, so if you go on to the next slide, I'm going to briefly introduce the Stark effect, which many of you are probably familiar with, but I just want to make sure we're all on the same page. This is just an electrochromic effect where we put a molecule that undergoes some type of electronic transition into an electric field and then measure how that transition is affected. And this is completely general for any type of transition throughout the electromagnetic spectrum. We're going to focus on the vibrational region of the spectrum. And the reason for that is the chromophore that we can use is just a diatomic oscillator. So it's essentially the smallest probe that we have for interrogating an electrostatic environment within a complex system. So the particular chromophore that I'm going to spend a lot of time talking about today is the nitrile. We find it convenient for a variety of reasons, which I'm not going to go into, but I'm happy to talk about it if you're interested. So basically, this nitrile has a fairly large ground state dipole moment, and when it is moved into an excited state, the bond changes in some way and that dipole moment changes. And it's that difference in dipole moment field which responds to the surrounding electric field of its environment, say an environment created by the dipole field that I just spelled out to you in a couple slides ago. If that field is oriented parallel to this dipole moment, that will result in an overall decrease in the absorption energy of the electron from the ground to excited state, and that's going to show up as a redshift in our vibrational absorption spectrum. If the dipole is oriented anti-parallel, that will increase the absorption energy, which will show up as a blue shift in our vibrational absorption spectrum. And so if you go, there are a couple other steps in this animation, but basically we start by measuring our absorption energy of our nitrile in some sort of background, what we consider a reference environment. And then by making a perturbation to this system, and it could be, for example, moving the position of the nitrile. It could be changing the chemical environment of the membrane. It could be changing any protein that it's interacting with. But by making a perturbation and then measuring changes in absorption energy surrounding that nitrile, we can directly map out the electric field to which that nitrile is exposed. And I think I have one more step of this animation, showing you the Stark effect equation, in which we have the vector of the dipole moment oriented with the vector of a field, which gives us a change in energy, delta E. And so the only unknown there is the field. Okay, so that was my only animation and we made it through. So looks like my slides are behaving themselves. Okay, so here's how we're going to actually do this experiment. So we need to put the nitrile into the interior of the membrane in some kind of controlled way. And we're going to do that by putting it onto a peptide. So the peptide sequence that I'm going to use for these experiments is shown here. We have 4 different peptides that are composed mainly of this alanine-leucine sequence. So it's very hydrophobic, completely insoluble in our buffer, but when it's part of a self-assembling lipid bilayer system, we're going to use vesicles in this case, it spontaneously inserts into the bilayer and forms a very well-structured alpha helix. And there are these anchoring units, these GPGG units at the end that anchor the head group in the membrane so that the peptide spans the Z axis of the membrane. And then the histidines are just there to make it teeny tiny little bit soluble just to be able to get this self-assembly process going. So what we're going to do, because these peptides are just made on a peptide synthesizer, is periodically along the sequence, we replaced an amino acid with cyanophenylalanine. And so this is what introduces our nitrile group at a known position in the membrane. And we just simply moved the position on the sequence where we put the cyanophenylalanine, and that's going to be how we move the nitrile in a systematic way. So the figure I have here shows a single peptide with 2 different nitriles. That's actually not how we do the experiment. We do the experiment with 4 different peptides, each one of which only has 1 nitrile. But we know exactly where that is and how it's oriented in the membrane. So next slide, please. And so this is just a CD spectrum. So just looking at the circular dichroism of the chiral peptide structure itself, showing that for all 4 of these peptides with our nitrile on it, they do indeed form a very regular alpha helix after being self-assembled in the lipid bilayer membrane. So, and we've characterized these vesicles in many different ways. We're very confident that the model structure that we're working with here is what I show you in a cartoon. And again, I'd be happy to talk about more of that if you're interested. So if you could go on. And here's an important thing to remember. So here I'm showing you 2 snapshots of 2 different nitriles in the membrane. And the first, this one that's labeled alpha LAX 25, this is where the nitrile is at its outermost position on the peptide. So this is closest to the head group region of the membrane. And what you can see is that as the cyanophenylalanine rotates around its chi-1 angle, which it has plenty of energy to do at room temperature, the nitrile, which is in blue at the very end of that ring, can be pointed in very different directions. So as it's in the snapshot right here, it's pointed down into the much more aliphatic portion, the hydrophobic portion of the membrane. But as it rotates, it can easily be pointed up into this region of the membrane that has much, much more water in it. So we would expect our experiments to show us that in steady state, this nitrile is experiencing very different chemical environments and therefore very different electric fields. Whereas in the snapshot on the right, I'm showing you this labeled position 16. This is where the nitrile is really smack dab in the middle of the membrane. And no matter how this rotates, it really should be seeing only the hydrophobic tails of the lipid. Okay, so again, that should show up in our spectroscopy. So next slide, please. So here is a summary of vibrational absorption spectra of all 4 of these nitriles on the different peptides. And what I want to draw your attention to is the figure in black, which is position 25. So that's the nitrile in the outermost part of the membrane. And then the spectrum in red, which is at position 16, which is in the middle of the membrane. And there's an almost 3 wave number shift between those 2 positions, which is a fairly large shift for us, compared to other work that we've done. And if we take so we know, looking at this equation on the bottom, we know the difference dipole moment of the nitrile. We've measured that independently. We know the angle A of the nitrile with respect to the perpendicular of the membrane normal, from our molecular dynamics experiments, simulations with our collaborator, Ron Elber, we measure the change in absorption energy, delta E. And so from all of that, we can back out that this 3-wave number shift gives us a field of 8 plus or minus 4 megavolts per centimeter in a DOPC membrane. Now, this number is interesting for a couple reasons. First of all, remember I told you when I was motivating this work that we were expecting to measure fields of, say, 1 to 10 megavolts per centimeter, and we're right within that range. We're right where we expect. The other thing I'd like to point out is this plus or minus 4 is not an experimental error, which is actually quite small. This represents the distribution of angles, A, from the molecular dynamics experiment. So like I said, at room temperature, there's plenty of energy for our cyanophenylalanine to rotate around Chi1. And when it does that, it's sampling actually a fairly broad range of electric fields. And we think that this is really important as we go further to learn how these electric fields affect membrane function, because it means that there are really quite extreme fields accessible at room temperature that then could lead to, lead along a chemical reaction pathway, which might not happen otherwise. So that was a fairly exciting result. So why don't you keep going? And then keep in mind that I've already told you that we should be measuring both changes in energy and also changes that represent homogeneous versus heterogeneous chemical environment of our chromophore. And so I'm showing you the same vibrational absorption spectra here that I just showed you, but now highlighting line width. And again, black at position 25 is the outermost position of the nitrile, which is in that top snapshot. Red is the innermost position, 16. And we see over 1.5 wave number difference in the full width at half max. And this represents the variety of chemical environments that these two chromophores are exposed to. And indeed, we see a fairly significant narrowing as the nitrile is moved into the interior of the membrane, just as we are expecting. So you can go on. And when we do the molecular dynamics simulations, of course, it's nice to have the snapshots to sort of see the system that we're working with and how it evolves over time. But we can also then use those simulations to make measurements of our system. And so on the left here, I'm showing you the exact same vibrational absorption spectra. And on the right, I'm showing you a calculation from our trajectories of molecular dynamic snapshots for just 2 of these peptide positions. In black is the outermost, in red is the innermost. And what you can, and what I'm doing here is that in each snapshot, we're measuring the electric field that's generated at the nitrile by the force field of the simulation. And what you can see in black is that this distribution is very broad, covers quite a large range of electric fields, whereas in red, the distribution is very narrow. And this is the equivalent of this difference in chemical environment that we see from the narrowing of the vibrational absorption spectra, which again completely conforms with our chemical intuition about how the nitrile is experiencing different environments as it moves down through the membrane. So let's go on to the next slide. Okay, great. It's all well and good to be able to make a direct measurement of this field in a membrane. That was really exciting. But what we're really interested in is how this can act, like I said, as a driving force for organization and function within a membrane. And so in order to do that, I think someone's unmuted. I'm getting a little bit of feedback. Okay, in order to do that, we need to move away from just this very simple DOPC membrane and start thinking about the fact that membranes are these chemically highly heterogeneous and quite diverse structures. And so the first thing we're going to do to take a step in that direction, a small step, is to add cholesterol. Okay, so basically what we did was repeat every single experiment that I've just told you about, but now adding increasing concentrations of cholesterol to our membrane. So if you go on to the next slide, So again, we have our alpha helical peptide labeled with our nitriles in the different positions. And now we're going to assemble our vesicles with DOPC plus cholesterol at known concentration. We did all of our characterizations again to make sure our system is the structure and chemical identity that we think it is. I'm showing you just a couple examples right here where where we had cholesterol at a concentration of 20 mole percent. We could actually go up to 40 mole percent easily. And so on the left, I'm just showing you more circular dichromosome spectroscopy to show we have our nice alpha helix. And on the right, I'm showing you now the vibrational adsorption spectra for our peptide now in this new chemical environment. Okay, but we really need to put all this information together. So if you go on to the next slide. So what I'm showing you here is, so I've been showing you vibrational adsorption spectra for 4 nitrile positions, but now we're just going to look at the difference between the outermost and the innermost. And so that's what I have plotted here on the y-axis. It's the shift in adsorption energy from the nitrile at its most hydrophilic versus its most hydrophobic position. And then on the x-axis here is just a function of the concentration of cholesterol in our DOPC going up to 40 mole percent, which is, by the way, completely relevant for mammalian cells. And what you can see is this very interesting bimodal behavior. When we increase sterile concentration, cholesterol concentration up to about 10 mole percent, we increase the magnitude of this dipole field. But then as we increase further up to 40 mole percent, we then decrease the magnitude of this field. And this was really interesting from our pursuit of the literature. When we first started this, we were actually quite confused what we would find. Some studies had found that increasing cholesterol concentration increases field. Some had found the opposite. And what we really found is it depended on what region you were looking at. It depended on your concentrations. But now the question is why? Why is it doing this? And so to try to tease that out, if you go to the next slide, we looked at a second sterol. There are many, many sterols that are biologically relevant in your body. One of them we looked at was ketocholestanol. And you can see that it differs from cholesterol only by the ketone at the 6th position of the sterol ring. Okay, so it differs by 1 atom. But we performed exactly the same experiments with the ketocholestanol. Here's some CD spectra and vibrational adsorption energy, but let's get on to the next slide where we can compare our results directly to cholesterol. So the black data here are exactly what I showed you a couple slides ago. And the red data are now the equivalent experiment for the second sterol ketocholestanol. So again, on the y-axis, we're looking at shifts in adsorption energy, which tell us about differences in the magnitude of the field that's being generated in these 2 membranes. Again, going up to 40 mole percent of the sterol. And whereas our cholesterol was bimodal, our ketocholestanol is monotonically increasing as long as we can add more cholesterol to the system. Okay, so completely different observation from cholesterol. 2 molecules that differ by a single atom. Okay, so what might be going on? So if we could go to the next slide. Again, remember way back when I was describing this experiment, I said that we're not only interested in shifts in adsorption energy, we're also interested in changes in line width. And so what I'm showing you now is for the cholesterol and the ketocholestanol. On the y-axis, we have the experimental full width at half max of the nitrile at position 16. So that's in the middle of the membrane. Again, going up to a concentration of 40 mole percent of the sterol. And if you look at the black line, you can see that it's steadily increasing full width at half max as concentration increases. That means that the chemical environment surrounding this interior nitrile is changing and becoming more heterogeneous as we add more cholesterol. Whereas the ketocholestanol in red is just kind of bouncing around with an experimental error, but not moving in any given direction. Okay, so the chemical environment of that nitrile is remaining the same. So what is going on here? If you go on to the next slide, this is a calculation we did from the simulations because our collaborators, Ron Elber and his postdocs, simulated a DOPC membrane containing either cholesterol or ketocholestanol at either 10 mole percent or 40 mole percent. And this is one slide where I wish I had control of my pointer to make sure I could walk you through all this data. But basically, black is the cholesterol, just as it's always been, and red is the ketocholestanol. And then the dashed lines are the low concentration, 10 mole percent, and the solid lines are the high concentration, 40 mole percent. So what I'm showing you right now is a calculation of a radial distribution function between a line defined by the helical axis, okay? So basically the vector that runs up the center of the alpha helix. That line and the cholesterol within a certain distance around it up to 28 angstroms, okay? So the distance is on my x-axis. And so peaks tell us the distances at which we find a lot of that molecule, and the valleys tell us where we're not going to find much of that molecule. And what you can see for the dashed lines for both colors, this is low concentration. We see very similar radial distribution functions, particularly at close distances. So the cholesterol and the ketocholestanol seem to be solubilized in the membrane in very similar ways at low concentration. But at high concentration in the solid line, we see two totally different behaviors. For the high concentration of cholesterol, we find quite a bit of it very near our nitrile. So that's that peak around, say, 8 angstroms or so. But for the ketocholestanol, we find that it seems to be separating itself from the peptide. And we only begin to see appreciable concentrations of it once it's pretty far away from our alpha helix out there at about 16 angstroms or so. So this is very different behavior of these two molecules in DOPC. And yeah, you can go to the next one, the next slide. And here I'm just going to step through what we think is happening as we increase the concentration of both of these sterols. So at 0 mole percent cholesterol, we have this fluid-like disordered phase. We're well above the melting temperature of DOPC. And our system is indeed randomly distributed in this two-dimensional plane. But as we go to higher concentration, say at 10 mole percent of cholesterol, we enter this so-called condensed disordered phase, where the vesicle is the same size, okay? We conduct our experiments by selecting for the size of the vesicle. But now the tails aren't moving around as freely, and so we have to put more lipids into the vesicle in order to get the same size. Well, each lipid brings in all of that water. That water is what's generating this field in the first place, and so this dipole field increases. But as we go to 40 mole percent, next slide, now we're in a condensed phase. The membrane has stiffened, and in order to put that much cholesterol into the system, we've had to displace lipids, okay? So lipids have left. They've left with all that water. Cholesterol has a molecular dipole moment, but it's much, much smaller than the phosphocholine with all those associated waters. And so now, as we add more cholesterol, this collective dipole moment is decreasing, the field is decreasing as we try to jam more cholesterol in. And that's very different from ketocholestanol, which is in this next slide right here, which never enters a condensed disordered phase. And moreover, what we see in the simulations, and it's hinted at this radial distribution function data I showed you a couple slides ago, is that the ketocholestanol is actually aggregating, and it's sequestering itself away from the peptide. And so again, we have, as we select for a particular size of lipid, we have more lipids per vesicle, and the dipole moment of all of these lipids plus the collective H2O is increasing. The dipole moment, the dipole field increases. Okay, so this aggregation of ketocholesterol gives us completely different effects of electric field at higher and higher concentrations than cholesterol. And this is really exciting because, you know, these molecules differ by one atom. I can't say it enough. And yet they result in totally different function of the structure of the membrane around this peptide. And it was really exciting that we could both measure that and simulate it. So if you go to the next slide, so we've just barely scratched the surface here. So I've kind of told you in a hand-wavy way that lipids are very diverse in biologically relevant environments. And here I'm giving you just a little bit more of a hint of that. At the top of this slide, I'm just reminding you about cholesterol and ketocholesterol. There are many biologically relevant sterols. One that we've started studying more and more is this molecule on the right, the 7-dehydrocholesterol, 7-DHC. And that differs from cholesterol just by the position of the double bond at the 7 position of the sterol ring. This 7-DHC is actually the final precursor to cholesterol in the biosynthetic pathway that generates cholesterol. And when the enzyme that does this final reduction of this double bond to take 7-DHC to cholesterol, when that is misfunctioning, this generates absolutely devastating consequences, particularly in the health of cells in the nervous system. And I don't really have time to go into why, but it's particularly devastating disease for children who've inherited a faulty copy of the gene that encodes 7-DHC. This is a primary cause of miscarriage, of really devastating developmental defects like anencephaly or microcephaly, so being born with either no brain or a very, very small brain, all caused by this single double bond difference between these two molecules. And right now it's completely not understood why. Is it because there's no cholesterol in these cells, or is it because there's too much 7-DHC and that it is doing something devastating in and of itself? And so we're essentially repeating these experiments that I've been explaining with now these other biologically relevant sterols to figure out how they affect electrostatics of the membrane. And then in the bottom part of this slide, I'm just giving you a little bit of the menu of the lipids that are found in human and other mammalian cells. They can, of course, differ by the chemical identity of the head group, by the linkage, by the length of the fatty acid, the number of elements of unsaturation, etc. You get this smorgasbord of possibilities. And the questions that we're now very interested in is how do different compositions of these various lipids between, say, a plasma membrane and a mitochondrial membrane affect the function of those membranes in ways that are incredibly important for the function of the whole cell. And even more excitingly, how can we use that as a way of targeting a specific membrane even when another is exposed to the same, for example, drug molecule that we want to deliver to a particular organelle. So that's a big area of our research in my group right now, which I fortunately am not going to have time to talk about. I'll go to just the next slide. And this is what I'm going to leave you with. So I mentioned at the very beginning the proteins, quote unquote, that we're working with are fairly small. We've been talking about just a simple alpha helical peptide. But in order to understand actual function of a membrane, we've gone even smaller. And we've spent a lot of time focusing on the single amino acid tryptophan. And it's kind of funny, my collaborator, Dr. Elber, calls tryptophan a peptide, which just amuses me to no end, but it's really just a single amino acid. And the reason we focused on tryptophan is, first of all, for a practical reason, it's a very handy fluorescent tag it's a very polar, you know, even if you think about the indole ring, I don't have a structure here that's readily available, but the indole ring is normally thought to be one of the hydrophobic amino acids, but actually it has a fairly long molecular dipole moment, and that's really critical for a Stark effect interaction with an electric field that's been generated by the membrane. And so the fluorescence peak of tryptophan turns out to be a really effective Stark reporter in and of itself. And moreover, the reason that we've started focusing on just a single amino acid or, you know, building up only very small peptides is we're interested in how these molecules can now transport from one side of the membrane to another. Everything that I've told you about so far is really electrostatics, the steady state electric fields generated by the structure of the membrane. But now we're interested in what happens when we put dynamics into the system. And again, this is just the dynamics of random fluctuations of molecules at room temperature or physiologically relevant temperatures. And what I'm showing you right here is a snapshot, again, from our molecular dynamics simulations of a single tryptophan molecule. So the green part of the molecule in the interior of the membrane, we're basically looking side on at the indole ring. And the backbone down here in red and blue is positively charged. And we control that positive charge either through pH or by chemical functionalization of the NNC terminus. And what I'm showing you is that this tryptophan can actually reach fairly deep into the interior of the membrane. And it's doing so in part because the indole ring is hydrophobic, but also, more importantly, the hydrophobic amino acid backbone is completing all of its desired hydrogen bonds with the with the head group region of the phosphocholine lipid. But in order to shepherd that indole ring into the membrane, into the interior of the membrane, excuse me, the exterior is actually deforming quite dramatically. The gold and blue right here are the phosphocholine head groups. The purple are the glycerol units, and you can see that they're actually moving, they're puckering into the membrane quite dramatically in order to solubilize that indole and the fatty acid. And they're even dragging water in with them in order to make this happen. So it's really exciting that, you know, we can do simple experiments to measure Stark effect changes of the membrane with the fluorescence chromophore of the tryptophan, and at the same time do simulations that give us really detailed snapshots that give us a mechanism of how this is happening and how this membrane is actually changing dynamically in order to carry out some sort of interesting function. So this is a major focus of my group right now, that we're just starting to publish more and more in, and that I'm really excited for moving forward. So I did have some other things to mention, but if you could actually just go to slide 53, I'm going to end here and just put up a conclusion slide. Yeah, you're gonna go
see
through
the slide number, but you can tell me which one that.
It's slide 53.
I don't see the slides number, but you can tell me which one is that. So this is
Here
one.
we go. Yeah.
Okay, cool.
Yeah, so this is just a summary of what I've been talking about our efforts to use vibrational spectroscopy. And I've only talked about working in the vibrational region of the spectrum in order to measure electric fields generated in membranes. We're now more and more using the fluorescence region, the electronic region of the spectrum, with tryptophan as our chromophore. And we're very, very interested in understanding how chemical diversity and chemical differences in membranes lead to function that's critical for membrane health. And again, that's far more important than just being a barrier between two spaces. So with that, I'm going to end. I've already thanked the people who've done the work, and I'd like to thank you all for your attention, and I'd be very happy to answer questions.
Thank you very much, Lauren. It was fantastic. I really enjoyed it. I hope so. Same for the attendees. Let me just a little bit rearrange the screen so I can see if there are any questions already in the chat. So Okay, I have plenty of questions actually before
Uh-huh.
people start asking. So maybe one very fundamental, if I got it correct. So you mentioned I really like that intro about distinguishing the transmembrane potential, which generates the field that people usually know in biology, I'd say, and neurophysiology. That also you mentioned the dipole field, which is interesting. Wasn't really well. I wasn't aware exactly about the terminology here, but I'm wondering here, the fundamental question I have. I mean, maybe fundamental for at least for me, is that you mentioned this dipole field is due to the dipoles of the phospholipid heads dipoles there, or what's the contributors there?
So the lipid does have a molecular dipole.
Right, exactly.
But what's really important is all of those water molecules.
Okay, so
I
see. So that's that makes it.
Mm-hmm.
Okay, just return to the slides. Okay,
yeah.
Because then I mentioned that it's actually the water, which is the main contributor. Okay. So that would mean if you would do the, let's say, lipid bilayer MD simulation, or let's say without the water, some people do in like in Waco in the holes for some time, you would not see those effects. I'm just curious. In the membrane?
Yeah, so you certainly wouldn't see the molecular cause of it. So you would have to make sure you're working with a force field that has incorporated that into its water model in some way.
Okay, I see. Okay. Another question I have is like maybe more, or actually maybe I can touch upon what you mentioned now in the MD, in the molecular dynamics simulations. How do you measure electric field in there? Because people sometimes use, the Poisson-Boltzmann calculation to calculate electrostatics inside, but how actually you do this for the purpose of matching with the experimental Stark shift?
Yeah, very good question. And this is not something we've done too much with in membranes. We've done a lot of it in our work with protein-protein interactions. So we've done essentially the exact same experiment using the nitrile as a vibrational probe of electric field. And now the deviation that we make to it is we just move the nitrile around, say, a surface that then docks with another surface of another protein. And we've done quite a bit of work to try to compare those to electrostatic calculations. On our own, we've primarily done it with APBS.
Okay.
We're doing more and more work using amoeba as the force field, and then just calculating field in amoeba, the same way I described it with our molecular dynamics simulations. And I will tell you that our absolute values tend to be way off. And in fact, often, not instructive. Trends, however, are usually pretty good.
Okay, this is actually something we are very interested in, because we are also doing MD simulations to try to understand how externally imposed electric fields to either an experiment or in MD can change the protein structure and then its function. So we are always asking the question, how far off are we in the values, absolute terms? And it's sometimes difficult to say, because we have something in experiments, but it's it's sometimes not directly comparable. So and also what we did recently, I don't think Jessica is not in the call. She was one of the colleagues in the team members. She was implementing amoeba just to include the polarizability of the electronic polarizability, because of for some spectroscopic predictions too. So I'm wondering, do you see any what is the trend of the effect? I know the answer. I would like to hear from you. If you take the non-polarizable force field and compare to amoeba, so is that what is the trends of the the electric fields you are getting?
Yeah. Okay. So this is
if there is a single direction, or is it more complex? I don't know. Whatever. You can elaborate on that.
So this is work that we haven't quite published yet. We're trying to desperately get it out the door. Amoeba has been a lot more challenging to work with than I realized.
Same here.
but Peng-Yu Ren, who is now really developing amoeba, the main developer of it, is here at UT, and even with his help, it's been challenging. Okay. So that's a very, very good question. We have a system. It's not anything I've talked about right now. It's actually GFP. And we're using GFP just because it's a well-behaved protein with a really well understood chromophore in it. Okay. And what we have found is that amoeba, if we use an amber force field plus APBS, again, absolute values are all over the place, but the trends are pretty good. And what we found is that for amoeba, the absolute values seem to be much, much closer to the experimental values, and trends are still good.
Okay.
So that's for one system. And, you know, keep in mind, you always have to think about what experimental data are you comparing to. Here it's a fluorescent chromophore that's really rigid, you know, surrounded by a protein, there's full of water, etc. Who knows how generalizable that result is going to be, but it's been really exciting.
Okay. Okay. So the amber force field, it was way off in a, well, let's say which direction? It was given a larger or smaller field?
So estimating fields that are far too high.
Far too high.
Yeah,
Okay.
I'm trying to, I don't have any slides I could pull up to show you because we're literally just now writing the paper.
Okay.
But
okay.
yes, it's overestimating magnitudes.
Magnitudes.
Mm-hmm.
Okay. Okay. That's interesting. That's really interesting. Okay. Yeah, we'll see. So we have to see how it will work for us because we haven't still then have the we don't have the amoeba outputs for our, let's say, our stuff, but I'm very curious if you compare that, because, you know, the point is, for example, in our computational experiments, so to call them, the MD simulations, we are seeing some effects, and we also see some trends with increasing field electric field strength. Like we are getting the effect, like if we have, for example, we are detaching one protein from each other, basically, for example. And this is, of course, electric field strength dependent, but nobody actually explored this kind of effects let's say on dependence on different force fields, or not including the polarizable ones. So yeah, that's really interesting. So whatever related to electric fields, let's say the molecular ones, will be out. I'm really looking forward to. If you have put your paper in the Hammer Hive or somewhere in preprint, it will be great to see.
Yeah, and what I'm really interested, what my group is really interested in is, you know, are there problems where you can use something that's really cheap and easy, right? If we could use amber as much as possible, great. But are there problems that you have to go to a more you know, more expensive model? But it's really important to know what's the best method for what question. Right. And what could I get away with something fairly easy to do versus, you know, amoeba, which is, you know, at least in our hands on our computers, takes quite a bit longer.
Yeah, for us, it's roughly the factor of 10, so 10 times longer.
Yeah. So, you know, yeah. So can we actually define what are those dividing lines where sometimes you're just going to have to do the harder experiment, right? And it's the exact same thing we do in the lab, right? When can we get away with something that's easy to do versus when do we have to, you know, pull out all the stops, right? And I think the question is exactly the same for calculations. One thing I would add, and again, I don't have any slides that I can pull up on this, but I'm really excited about it. Keep in mind the data we're comparing to are spectroscopy. So we're looking not just at overall changes in energy, we're also looking at changes in line width. And what we have found is that using amber plus APBS, even if our absolute values are widely off, if we go through our molecular dynamics trajectories and bin all of our snapshots, so essentially calculate a field in every snapshot and bin them, we can actually replicate spectral features in line width. We can replicate things like shoulders, which indicate that there's a large population that's sort of near the main population, but not quite on it. And that's also quite exciting because it means, okay, I'm not going to get absolute values, fine, but I can still understand shapes, and that gives me molecular information.
Okay, that's great. So at least the amber is useful for that. Well, you touch upon the line width. I was curious, we are not doing just stark spectroscopy, but I know it's a powerful tool for especially for measuring the molecular fields, as you beautifully showed. I'm curious, what are the challenges there in terms of interpretation of the line widths, right? Because there are different broadening mechanisms, and you would like to have the one which is related to the, well, local electric field. But what are the other, let's say, confounding factors which can, you know, make it?
I could give a whole other talk.
It'll be great.
The principal thing that we have to worry about with the nitrile as a probe in particular is hydrogen bonding.
Okay.
Because the lone pair on the nitrile is a good hydrogen bond acceptor. And so if you think about the nitrile has that triple bond, right? So it has this huge pi cloud in the middle of those two atoms. As it turns out, that pi cloud is a good hydrogen bond acceptor itself.
Okay.
So we can have hydrogen bonds that are coming end on or side on. And as it turns out, they have two different effects on adsorption energy, but these are quantum mechanical effects, right? So they have nothing to do with electric field.
Okay.
That's partly why we have always been so insistent on doing molecular dynamics simulations so that we can quantify hydrogen bonding.
Okay.
Experimentally, we test it through changes in vibrational spectra as a function of temperature. Because as you change temperature, what happens is you just kind of change the tumbling rates at which two neighboring molecules are interacting with one another. And that changes, let's say I have a hydrogen bond that's end on my nitrile right here, but I heat it up and now it's tumbling around a bit more. That changes the adsorption energy of the nitrile. And as long as we know what that effect is, we know the strength of that hydrogen bond, and we can incorporate that into an electrostatic interpretation once we've essentially zeroed out hydrogen bonding.
Okay, I see.
Yeah, but that's a major issue to worry about.
Okay. Yeah, I see. So that's the major one. Of course, I can imagine there are plenty of like technical ones, but probably it's something you can, once you know the stuff, you can sort out.
There are technical, the nitrile is nice to work with because it adsorbs at about 2200 wave numbers or so. And if you think about a background of a protein or a membrane or a peptide or whatever, and you think about all those other functional groups that have stretches and bends and everything throughout the vibrational region of the spectrum, there's a window between about 2000 and 2500 wave numbers that's fairly free, and the rest of it is just completely full of the protein background. And so a very practical reason for using the nitrile is because it's really in the only free region of the spectrum.
I see. That makes sense.
Yeah.
Okay. I'm wondering if people are a bit shy, or actually you are the first speaker who are in our series who is speaking about Stark spectroscopy. So I think it's quite illuminating for the people who are coming mostly from, let's say, some biophysics, some of the engineering, biomedical engineering. So I think it'll be great that we have this recorded so people can refer back to that in the context.
Yeah, no, that makes me really happy because I think for a long time, many have thought of this as sort of an esoteric technique that only a specialist can do. And my group has done a lot of work to try to break down that myth. I would really love to see more people trying to apply this to their own problems because it's such a direct measurement of field. PKA, right? PKA is great for whatever. There's very little interpretation required here.
Oh yeah. That's true. So that brings me to the last thing that you mentioned, using the tryptophan as a probe. So you mentioned that you are now kind of probing the fluorescence and its shift as a, let's say, probe of the electric field. So I can understand this works. Typically, what I remember, what I know from the physical chemistry textbooks and also from experiments, tryptophan is said to have a blue shift or red shift depending on people say macroscopically effective polarizability or permittivity of the surroundings, right? But it's actually electric field when people want to go to the detail. That's how I understand that.
Yes.
So how do you work with the tryptophans? You're looking on exciting in, I don't know, 290 or 80, or depends on what works for you, and you are looking on what are the shifts you are seeing, let's say, in the membrane compared to when it's in the solution.
Yeah, actually, you could go to slide 43.
Sure.
And I could show you that explicitly. So we're exciting around 300.
Nice.
Yeah, so this is the fluorescence of tryptophan. We're exciting around 300, so down here. And this is just a simple proof of concept. So this is tryptophan dissolved in hexanes, and it's emitting around 340. And then when we put it in either, just HPLC grade water or buffer, it shifts quite dramatically to the red, up to about 375 or so.
Okay, this is only due to the proton concentration or the pH basically.
It's purely due to we call it the reaction field that's set up around the tryptophan from either a low dielectric hexanes or a very high dielectric water.
Okay. Yeah. So this is now what you consider to be a tool to be used, let's say in probing in okay.
Yeah.
So, but terminology wise, people don't call this Stark shift typically, right? Or do they? I mean, in your field, I mean.
Yeah, absolutely. We call this a Stark effect. Absolutely.
Okay, it's called a Stark effect too. Okay, I mean, physically it's the same thing, but people usually don't mention that in directly, because I usually, when I hear Stark effect, I'm thinking about infrared spectroscopies and shifts of the peaks there. But this is actually, well, yeah, that Stark field, whatever electric field causes as a shift, it could be called a Stark shift.
Exactly.
Okay, I see. Okay, cool.
Mm-hmm.
And there are here any? I mean, the tryptophan is great, right? Because it's a natural, I mean, indigenous probe. So it's maybe a little bit easier to interpret. But what are the challenges here? What you see in terms of, let's say, and again, calibrating and speaking about some local electric fields, how is it being done?
Yeah, the main challenge experimentally of tryptophan is it's nice because it is one of our natural amino acids. It does have a fairly weak molar absorptivity.
Okay.
Compared to, for example, dyes that like the microscopy community is very familiar working with. So you have to work at concentrations which may be difficult to achieve for some experiments, because you have to increase concentration. We've started recently working with another dye, Danzel, which is no bigger than tryptophan, and it has a nice big sulfate group on it.
Okay.
Which increases its dipole moment and its molar absorptivity. The problem is it's not a natural amino acid, so now you have to worry about how you get it into your system. And you also have to worry about introducing this new sulfate into, a peptide or whatever that you put it on, and what does that do also to the rest of the structure?
Okay.
So there are other options. Tryptophan's been very convenient for us. The concentration issue hasn't been a problem so far.
Okay, I see. Cool. I've seen a raised hand here, notice, but I think the already disappeared. So probably, well, we are some little bit over, let's say 5 p.m. and 10 minutes here, so maybe people should start leaving. But let me check my notes. So, okay, one more which should also touch upon, and something which we're curious about. As our research team has this name of bioelectrodynamics. So we're trying to see if let's say, not only the electrostatic effects in molecules, what could be taking place, but also if there are some dynamical effects of, let's say, fluctuating fields, and if that has any role in a biology or interaction between the molecules. So you mentioned something like that in your very last slides. You described in your talk that you could maybe elaborate on that. If you see something like well, there is always some Brownian motion, thermal motion. So there'll be the sources of the field are fluctuate. That means atoms, ionized groups, are fluctuating too. So do these things just average out, or is there something related to the time scales of these fluctuations which could matter? What do you think about that?
So in the experiments that I showed you with our nitrile in the membrane, everything was steady state. So it takes, 5 minutes or so to collect the spectrum. And so you're just seeing, yes, everything is averaged out. The different populations that were transiently apparent show up in line width. Okay. But we can't individually measure them. However, in a time resolved experiment, you could absolutely do that.
Okay.
So time resolved infrared spectroscopy, infrared chromophores usually only live for a few picoseconds. And so you're looking at fluctuations that happen at, say, the picosecond, maybe the 100 picosecond time scale. Not a lot interesting in biology happens in 100 picoseconds, right? Biology is kind of slow, but you can absolutely see random motion.
Okay.
The really nice thing about working with chromophores like tryptophan or chromophores that are working in the visible region of the spectrum is that they tend to have much, much longer lifetimes.
Okay.
Now getting into nanoseconds, hundreds of nanoseconds, some can even go into microseconds. And so, there's a lot more that a protein or a membrane or something can do in that time span that you can actually watch with your excited chromophore.
Okay, I see.
So
okay.
Yeah, so everything that I've talked about is completely transferable to a time-resolved experiment. The question is, what's the time?
Yeah, I see. Okay, so now I see the raised hand from Vladimir Berest. I'll try to unmute and let's see if it works.
Thank you.
Or
probably I just missed a part of the lecture, but my question is it possible that this dipole field could be a driving force for the formation of lipid domains, like redistribution of different sorts of lipids and to form domains?
That is absolutely our hypothesis. Yes.
Thank you.
So stay tuned.
do you want to elaborate on that, Lauren? If it's like or it's just something you are thinking about?
Yeah, no, I think the question articulated it very well, that this provides a driving force for causing some things to interact with each other in the membrane. So for example, a common motif that you see in membrane-bound proteins is the alpha helix. And in fact, these alpha helices tend to bundle into groups that look very, very similar for many different types of proteins. Well, an alpha helix, I haven't talked about it, but an alpha helix has a helix-long dipole moment,
right,
generated by the amino acid backbone.
Right.
Okay, and so that dipole moment is in the field, right? It's now in this field that's been generated by the membrane. And this could be a driving force for causing these aligned dipole moments to come together and aggregate in this complex reaction field environment, but not allow other types of structures or other molecules or something that's just only has a few amino acids tethered in the membrane, but isn't fully immersed in it. So I think absolutely that this provides an energy landscape, which is going to funnel into particular structures, things that we like to call domains.
Okay.
And this is really why, I've said a couple times that we're really excited about moving away from model membranes that are just a single amino acid, a single lipid or a single lipid plus cholesterol. And we really want to start to explore the vast menu of lipids that we know are in biologically relevant membranes, because we think that one of the most important reasons that you would substitute one lipid for another is to change the field environment, right? Okay. Which would then organize one particular type of structure as opposed to another.
I see. There's one more question from Vladimir. If you can unmute.
Thanks. So my other question is, what do you think if along with lipids, we add some carbohydrates, like if we're gonna build a model from, say, lipids with sugar tails, would impact somehow the distribution of fields and probably affect the energy required to separate lipids and probably separate some proteins in this say, hairy surrounding?
I absolutely agree. When you're looking at just the lipid itself, there's so much more chemical complexity within an actual living cell. And so absolutely, and really trying to build piece by piece, start simple, figure out the experiments, and then add complexity. I've only talked about adding different kinds of lipids or things like sterile molecules, but absolutely add the sugars. And our synthetic colleagues are getting better and better at making really complex lipid sugar constructs. And so we could think about doing those experiments in some sort of systematic way. But yes, absolutely, they're all a part of this really, I think that's what I put in my slide, complex environments, right? And we need to account for all of it. So yeah, there's only more to do.
Yeah, absolutely. There's definitely a huge, almost infinite, broadly speaking, let's say, possibilities where to go. So what do you think will be, you kind of highlighted it, but what do you think is a way to go for, absolutely, there is something you want to pursue in your lab, but if you, let's say, would have a, much greater resources, or you could be like steering the research field where to go, what do you think would be the let's say, that kind of message to the people to what's the most interesting or exciting thing to go in this field?
Very good question. So when I've talked about being interested in different types of lipids, I've really talked about differences within a given cell, looking at organelles. But going back to just cataloging what lipids are present in what cells, there's now also quite a lot of information in how cells change over their life cycle, but also as a function of disease state. For example, we know that cells that have metastasized have lipid compositions in their plasma membrane that are quite different from a healthy counterpart, even to the point that just a mass spec of the lipid profile can be used to identify the disease versus the healthy cell.
Okay.
And so this is now becoming more and more common in diagnostics. Like, let's say you're a surgeon. I have a colleague here at UT who does sort of spatially resolved mass spec on living tissue. And so let's say you're a surgeon removing a cancerous tumor from a body. Well, you want to make sure you get all of it. So if you could do mass spec on the spot to know when you've cut it all out and you're only seeing healthy tissue, that could be very helpful. Let's actually take that a step further. Drugs that target that are used to treat cancer are often devastating to the people who take them. They have devastating side effects because they target all cells kind of indiscriminately. So what if we could develop some sort of therapeutic strategy that not just targets a cancer cell as opposed to a healthy cell, but a cancer cell in only one particular type of tissue, as opposed to all the other tissues that drug might be exposed to. So I think there's some really profound steps that can be taken in taking this knowledge and applying it to, say, strategies for treating disease, if we understood what the mechanism that causes a cell of a particular lipid composition to act differently than a healthy counterpart. So there's a lot of very fundamental work that needs to be done, but the potential applications, I think, are really quite exciting.
Yeah, that's really interesting. Yeah, that's this perspective which you outlined. That's great. Okay. Good. So let me see this chat. I don't see any more questions. So I think people are already exhausted, positively, I believe.
Well, it's the end of your day and the beginning of mine, so I'm full of energy.
Yeah, well, I mean, this still really was exciting for me, so I really enjoyed that. Hopefully, same for our attendees, which made a few tens today, so that's great. So I think I should now thank you, Lauren, for a great talk. I really enjoyed that, and I hope that you'll be also interested in our future webinars, which will follow. We have a mixture of things ranging from biophysics to micro nanotechnology, all related somehow to electricity or electromagnetism on the scale of molecules and cells. So definitely it'll be great if you can also follow up and also forward to other people. So thank you, Lauren. It was a really great talk. And have a good day, and hope to see you somewhere in person when the things will be
one day
possible.
Well,
so
thank you
very much. And I'm stopping now the recording. So thanks to everybody for the attendees, and hope to see you soon in other webinars. Bye. Have a good day.